This part 2 of my 1.3 cardiovascular system assignment. To just recap last video, this is the structure and makeup of blood, and this can be, we can get to this by centrifugation, which is this process here. And we got to plasma, which makes up 55% of the blood structure. It's 90 to 92% water, and as you can see, it's a straw-coloured liquid. It contains electrodes, blood plasma proteins, hormones, and its functions include maintaining optimum body temperature throughout the organism, and it maintains pH level of the blood and blood tissue so that the cells within it can thrive. It also maintains the ideal balance of electrodes in the blood and the tissue. The next thing we're going to look at is the red blood cells, which make up 45% of the blood structure. The lifespan of these are about 120 days, and they're a red pigment which also has no nucleus. So there's room for more hemoglobin, more CO2 within these. And that's its main function. Also, it contains hemoglobin. Obviously, it's a small size, flat dish shaped, and the fact it doesn't contain a nucleus means there's more room for the hemoglobin. The functions include the hemoglobin, which absorbs oxygen in the lungs and releases the oxygen to the rest of the body. And then looking at the second final thing is the white blood cells, which make up less than 1%. These literally have a lifespan of a few hours, and they're used as transportation to infected areas. They produce antibodies and that to destroy the harmful infection, and they can engulf any infections that are there. So they make up a major part of our immune system in order to help the body fight infections. And the last part is the patellas. Patellas are cell fragments. They're also a disc shape like the red blood cells, and they have a diameter of 2.4 micrometers. All similar to the red blood cells, there's also no nucleus, but these have a lifespan of about 5 to 9 days, and their function is to facilitate blood clotting and to prevent the loss of bodily fluids as well. Right, moving on to the blood cells and the structure and functions of the blood vessels. Looking at the arteries to begin with, as soon as the blood leaves the heart, it goes straight to the arteries. The arteries are a thick, muscular, elastic wall, which this thick elastic layer allows for expanding and recoiling. It's controlled by the sympathetic nervous system, and the muscular tube lined by smooth tissue is the thickest muscle wall, which is this muscle wall is formed by 3 different muscle tissues: the intima, the inner layer lined by smooth tissue, the medium, which is the layer that allows the arteries to handle the high pressure, and the external, which is a tissue that anchors the arteries to the nearby tissues. And obviously the arteries have the second largest diameter of lumen, with only the veins having a bigger one. Blood obviously comes on here through the arteries up very incredibly high pressures and velocities, as it's just been ejected from the heart, and the arteries carry these away, and and yeah, these carry them on until they reach the arterioles. The arterioles don't have any the next thing. They don't have any fibrous tissues or elastic tissues. However, they have high levels of smooth tissue, and they are in the middle as far as mean diameter and mean thickness is concerned. As mentioned before, vasoconstriction happens here. So this is where the blood constricts and the flow of blood is reduced, retaining the body heat and increasing vascular resistance. And the skin is obviously paler out here, and opposite to the vasodilation, where the skin will become reddy coloured when the blood is pumping to the where a higher flow of blood is allowed through and able to reach the surface of the blood. Next to the capillaries, these have no smooth tissue or elastic tissue, and they are the smallest. They have the smallest mean thickness, so that diffusion can happen easy, so that the oxygen and carbon dioxide can pass through the cell walls very easily. They have the smallest diameter, and they're drained by venules. However, there is despite the thin cell walls, there are a large surface area for capillaries, as there are so many, because the gas exchange will be able to happen over a greater surface area, so that makes it easier for this process to happen. The capillary bed surround the organs, and the entrance to the bed is regulated by the precapillary sphincters, which is a band of smooth muscle tissue. The relaxation of the sphincter allows for increased flow, and constriction of the sphincter decreases flow. So this will obviously slow down the rate of diffusion. After the blood and deoxygenated blood is being carried away from the muscles, venules then become evident. Venule walls have this also have 3 layers, and these are these literally just are just small blood vessels that allow for deoxygenated blood to return from the capillary beds to the largest of the blood vessels, the veins. And the veins are walls also consist of thinner 3 layers, but however they're thinner and they're less elastic, so they as they don't need to expand because they're not going under the same sort of pressure that the arteries will be. Also they have pocket valves which stop any backflow from the because obviously the blood is travelling upwards when travelling towards the back towards the heart. So to stop any backflow, the pocket valves will open, and they will be forced open, and they will start to fill once the blood starts to fall backwards with gravity. The veins also, obviously, main function is to return blood to the body, and this could be deoxygenated. If it's oxygenated, it's returning the blood to the left ventricles, and this through the pulmonary, left atrium, through the pulmonary veins, and the deoxygenated blood is taken back by the veins to the right atrium.